Good afternoon, friends. I'm Solomon. I'm here to present the second part of the chapter of the general biology course. In this part, proteins, nucleic acids, vitamins, minerals, and water are going to be discussed. So please don't forget to like, share, subscribe, and comment this channel. And let us continue to protein part. Well, proteins are the third macromolecules that are required large amounts for our bodies. Proteins are made up of different amino acids. That means they are polymers of amino acids. These amino acids are joined together by a type of covalent bond called peptide bond. Amino acids are compounds which are formed from carbon, nitrogen, oxygen, hydrogen, must. Meaning all amino acids have these elements or these atoms in must. But some amino acids may also have sulfur in the other molecules. But all amino acids have these four elements in common. There are around 20 amino acids are found in nature, or well-known amino acids in nature are around 20, and they have the same general structure in common. For example, this is the general structure of all amino acids. This is central carbon. Central carbon is bonded with 4 different molecules. For example, the first molecule is amino group. Amino group is bonded with the central carbon, or you can say it is alpha carbon. And also this is a carboxylic acid group, which is attached with the central carbon with another side, and hydrogen is there, plus the R group or variable group is attached with central carbon. Therefore, all amino acids have central carbon, amino group, carboxylic acid group, and hydrogen, but their difference is on the R group. I'll show the different amino acids in the next slide. The only difference of these 20 amino acids is on the R group. Whenever 2 amino acids are bonded to each other with peptide bond, dipeptide molecule is formed. Here, the peptide bond is formed between the carbon in carboxylic acid group, the carbon in the carboxylic acid group, and the nitrogen of the amino group of the second amino acids. Whenever 2 amino acids are joined together by peptide bond, the peptide bond is formed between the carbon of carboxylic acid group, carbon of the carboxylic acid group of the first amino acid, and the nitrogen of the amino group of the second amino acids. So immediately this peptide bond is formed. Immediately water molecule is lost, because here in carboxylic acid group there are 2 oxygen atoms. 2 oxygen atoms and 1 hydrogen atom are there. 2 oxygen atoms and 1 hydrogen atoms are there. But here 1 oxygen atom is left. Therefore, 1 oxygen atom and 1 hydrogen atoms are lost. 1 oxygen atom and 1 hydrogen atom are lost from the carboxylic acid group of the first amino acid. Whereas the amino group has one nitrogen and two hydrogen atoms. Therefore, one hydrogen atom is lost. Plus here, one hydrogen atom from amino group of the second amino acid. Therefore, immediately OH plus hydrogen gives what? H2O, water. Therefore, in this manner, water molecule is lost from when two amino acids are joined together by peptide bond and the dipeptide molecule is formed. Similarly, water molecule is lost. This is the process is known as condensation process. These are structure of amino acids as I have said already. Their differences on the variable side chain or the R group. Therefore, here we can identify each of these amino acids based on their difference. For example, glycine and alanine are simple amino acids. They are simple amino acids because their R chain contains hydrogen and CH3, which is methyl group. So they are simple amino acids. For example, serine and threonine. Serine and threonine have OH group on their side chain, so they are hydroxyl amino acids. You can say that hydroxyl amino acids. Still, there are branched amino acids: valine, lysine, and isoleucine. They are branched amino acids here. Valine is here. Lysine and isoleucine. Their hydrocarbon chains are somehow branched and longer. Still, there are amino acids that contain sulfur. For sulfur-containing amino acids are cysteine and methionine are sulfur-containing amino acids. Cysteine and methionine. Here is cysteine and here is methionine. They contain sulfur in their R chain or the variable side chain. Therefore, they are sulfur-containing amino acids. Still, there are amino acids that contain amide group. These are amide group containing amino acids are asparagine and glutamine. Asparagine and glutamine are amine containing amino acids because this NH2 is amide group. For these amino acids are amide containing amino acids. Still, there are amino acids that contain one additional carboxylic acid group. Already, all amino acids have one carboxylic acid group, but these amino acids, the aspartic acid and glutamic acid, even their names are acid because they contain additional carboxylic acid groups. Therefore, aspartic acid and glutamic acids are amino acids that contain additional carboxylic acid groups. So they are acidic in nature. Whereas amino acids with one additional amino group, they are lysine, arginine, and histidine. These have additional amino group. They have additional amino group. Lysine. Where is lysine? Lysine here. This is lysine. Contains one additional NH3+, that is amino group. arginine is also contains one additional amino group. Still, histidine also contains one additional OH. The amino group. Therefore, these amino acids are basic in nature. They are basic in nature. There are amino acids which have heterocyclic molecules. These are tryptophan is an example. Tryptophan is an example which contains heterocyclic side chain. This is heterocyclic. At least there are two cycling molecules are there in their side chain. Therefore, this tryptophan in tryptophan, the side chain contains heterocyclic molecule. Another amino acid which has special arrangement is proline. Proline is amino acid. It is R chain arrangement, or the variable side chain arrangement is somehow different from the other amino acids. Its the central carbon, or the alpha carbon, is joined with the R group, as well as the R group is connected with the amino group of the amino acid. So its arrangement is somehow different. Here you can see this is alpha carbon or central carbon. This R carbon is connected with R groups or the variable side chain. However, this variable side chain is connected with the amino group of the amino acids. Therefore, this amino acid proline has some different arrangement, different structure than other amino acids. So it is amino acid, you can say it. Aromatic amino acids are there also, because they contain, or their variable side chain contains amino molecules, or aromatic molecules. For example, phenylalanine and triosine are or have such arrangement. Here, triosine has the aromatic side chain. Still, phenylalanine also contain the aromatic side chain. So, because of the variable side chain, these amino acids have different in nature, different in their property. Well, this is structural level of protein. Protein molecules pass through different structural level to be matured and finally reach as a functional state. Therefore, the first level is primary structure. In primary structure, sequences, numbers, and the types of amino acids are considered, because each protein molecule has special sequence, number, and the types of amino acids. Therefore, in primary structure, only the sequence, number, and the types of amino acids are considered. The sequence, number, and the types of amino acids in protein molecules are determined by gene or DNA. Therefore, once this special number, type, and arrangements or sequence of amino acids are done, then after it passes to the second structural level. This is secondary structure. So in secondary structure, side chains of amino acids or different amino acids of such polypeptide molecule, they react to each other. They react to each other and they make bonds such as hydrogen bond, ionic bond, hydrophobic interaction, van der force. These all forces and the bonds will be there to join the side chains of different amino acids in polypeptide molecule. Therefore, as secondary structure, the arrangement will be either in the alpha helix structure, or it can be in the beta plated sheet form, either in the alpha helix structure form, or it may be in the beta plated sheet form. To show the figure, the alpha helix form, it looks like here, simply tear-like structure. It looks like tear-like structure, but in beta plated sheet, some sheets are formed. Therefore, at this stage, still the protein molecule is not functional protein. Therefore, it must pass to the tertiary structure. In tertiary structure, further such reactions or such bonds, such as hydrogen bond, ionic bond, hydrophobic interaction, van der force, these all force and bonds will be carried out further. Then tertiary structure will be formed. At this stage, it shows three-dimensional foldings. It looks like an tangible object. Most protein molecules at tertiary structure, they are functional at this stage, but still some protein molecules need additional structural level, which is known as quaternary structure. In quaternary structure, different mean two or more polypeptide molecules that reach a tertiary structure will assemble together. For example, if this is one polypeptide molecule, and if we change the color, if this is another polypeptide molecule, and still if this is another polypeptide molecule, and another, if this is another polypeptide molecule, therefore this protein molecule has 4 polypeptide molecules that are assembled together. For example, hemoglobin is such type of protein molecule. It has 4 polypeptide molecules that are assembled together. Therefore, at quaternary structure, 2 or more polypeptide molecules are assembled together, the functional protein. Therefore, these are the 4 structural levels that protein molecules can pass and reach to their mature and functional levels. Okay, here is functions of protein. Protein has a number of functions for our body. For example, around 15% of our body mass is made up of protein. For example, our skin, our muscle, our skin, our hairs are all made up of proteins. Around 10,000 different protein molecules or protein types are found in our cells, and they are function for example, they are functional in structural support. For example, cytoskeleton, which is a protein network, which provides shape and size of cells. Therefore, it supports these protein molecules can structural support for the cells. And still, these protein molecules can used as a transport mechanisms. They can transport different substances in and out of the cell, especially transport proteins, which are found in the cell wall. In the cell membrane of cells, used to transport substances in and out of the cell. Still, proteins are important in the communication purpose, or they are used in communicating signals. For example, when protein molecules are joined with carbohydrate, glycoprotein molecules are formed. Therefore, these molecules are very important for cell-to-cell communication and for cell-to-cell recognition. Still, these proteins are very important in speeding up of chemical reactions. Almost all chemical reactions that occur in our body needs enzymes which catalyze such chemical reactions. Therefore, all enzymes are proteins. Without enzymes, life is impossible. Therefore, these proteins can speed up chemical reactions of living organisms. Proteins can control cell growth. For example, growth hormone, which is a protein molecule that control the growth of organisms. Still, proteins can defend our body against different disease-causing agents. Such proteins are known as antibodies. So, proteins have a number of functions for living organisms. Okay, this is nucleic acids. Nucleic acids are DNA and RNA, which are genetic materials that store and transmit genetic information from parent to offspring. These nucleic acids are polymers of nucleotides. Carbohydrates are polymers of monosaccharides. Lipids are polymers of glycerol and fatty acids. Proteins are polymers of amino acids. These nucleic acids are polymers of nucleotides. So there are two types of nucleic acids, as I've already said: DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid. Nucleotides are formed from three main components. These nucleotides, which are monomeric units for nucleic acids, are made up of phosphate, nitrogenous base, as well as 5-carbon sugars, which are pentose sugar. Here, pentose, a phosphate group plus base plus sugar gives nucleotide, one nucleotide. When these three components or three molecules are joined together, one nucleotide molecule is formed. Here you can see this is nitrogenous base. This is nitrogenous base, which is joined with sugar. The sugar is pentose sugar, 5-carbon sugar. This one is phosphoric acid or phosphate group. Therefore, when these three molecules are combined together, they make a single nucleotide. Then after, in this form, once nucleotides are formed, they are joined together to make nucleic acids. Anyways, nucleotides are made up of these three different components. This is the structure of cyclic form of ribose sugar. Ribose sugar is a 5-carbon sugar and has such cyclic form. The difference between ribose and deoxyribose is the number of OH group on carbon number 2. Here, D2-deoxyribose means there is one oxygen is lost from the carbon number 2. Therefore, the difference between ribose and deoxyribose is the number of oxygen which is lost from carbon number 2. This is nucleotide 4, which is made up of nitrogenous base, which is joined with 5-carbon sugar. Specifically, this is deoxyribose sugar because there is no oxygen on carbon number 2. And this 5-carbon sugar, which is deoxyribose sugar, is joined with a phosphate group. So these 3 molecules are joined together to make one nucleotide. Here, when nitrogenous base is combined with a sugar molecule, immediately nucleoside molecule is formed. Here, base plus sugar give a nucleoside. And when nucleoside joined with a phosphoric group or phosphate group or phosphoric acid, then nucleotide is formed. The 5-carbon sugar in DNA is deoxyribose, whereas ribose is for RNA. There are 5 nitrogenous bases are found in nucleic acids. These are adenine, thymine, guanine, cytosine, and uracil. These are 5 amino acid, nitrogenous bases which are found in nucleic acids. Adenine and the guanine are purine base, which this purine base is base with 2 rings, whereas thymine, uracil, and the cytosine are pyrimidine base. Pyrimidine base. Pyrimidine bases are with a single ring. Here, purine bases are adenine and the guanine. Adenine and the guanine they have double ring. But pyrimidine bases are cytosine, thymine, and uracil. They have a single ring. This is the difference between pyrimidine and purine base. These 4 nitrogenous bases are found in DNA. Adenine, thymine, guanine, and cytosine are found in DNA, whereas uracil replaces thymine in DNA. Therefore, in RNA. Therefore, RNA contains adenine, guanine, cytosine, and uracil bases. DNA is a double-strand molecule, whereas RNA is a single-strand molecule. In DNA, always adenine is paired with thymine and the guanine is paired with cytosine. In RNA, adenine is paired with uracil and the guanine is paired with cytosine. Here, RNA is a single-strand molecule, but in some cases, especially during transcription, for example, it may be found in double-strand form for a short period of time. At that time, adenine is paired with uracil and guanine is paired with cytosine. These are some difference between DNA and RNA. DNA contains deoxyribose sugar, whereas RNA contains ribose sugar. DNA is double-strand molecule, but RNA is single-strand molecule. DNA contains adenine, thymine, guanine, and the cytosine bases, whereas RNA contains adenine, uracil, guanine, and the cytosine bases. In DNA, adenine is paired with thymine and the guanine with cytosine. In RNA, adenine pairs with uracil, whereas guanine is with cytosine. So these are main difference between DNA and the RNA molecules. Okay, vitamins are the fifth biomolecules. They are organic molecules like that of carbohydrate, lipids, protein, and nucleic acids. However, they are small. They are required in small amount, so they are micronutrients, not macronutrients, because they are required or they are needed in small amount for the metabolic activities of the body. Many vitamins can be used as a coenzymes. These coenzymes are molecules that help the enzyme functions, because these enzymes are proteins. All enzymes are proteins, but in most cases, this protein part of enzymes alone can't be a functional enzyme. Rather, it needs additional non-protein molecules. Therefore, vitamins can be used this purpose, meaning this is a protein part of enzyme plus non-protein part of the enzyme, which is coenzyme. For example, coenzyme means for this case is vitamin. Then after, functional enzyme will be produced. Functional enzyme that catalyze chemical reaction in appropriate way. Functional enzyme. This is protein part of enzyme, and this is non-protein part or the coenzyme part of the enzyme. When the two combine together, functional enzyme is produced. Therefore, this non-protein part of an enzyme is known as coenzyme. So vitamins can be used for this purpose. Plus, enzymes can prevent deficiency disease. These deficiency diseases are diseases that are caused by a lack of balanced diet. For example, lack of vitamin A results in night blindness. Lack of vitamin D results in scurvy. Lack of vitamin D results in rickets, and so forth. Therefore, vitamins can prevent us from different deficiency diseases. Vitamin can have or can be produced from various sources. For example, this vitamin D. Vitamin D is made or produced in the cells which are found in the skin. These cells in the skin, when they are activated by sunlight, they produce vitamin D. Actually, sunlight can't give us vitamin D directly. Rather, sunlight activates cells in the skin, then the cells produce vitamin D. Some B vitamins and vitamin K are produced by bacteria, which are found in the large intestine. Most vitamins are obtained from balanced diets, especially green leaves and different fruits can provide us different vitamins. Vitamins are categorized or grouped based on their solubility. Some vitamins are fat-soluble vitamins. Some of vitamins are fat-soluble vitamins, and they can be stored in the small quantity in the liver and the fatty tissue of the body, because they are fat-soluble and they can be stored in the liver and fatty tissues of the body. For example, vitamin A, vitamin D, vitamin E, and vitamin K are fat-soluble vitamins, and they can be stored in the liver and the fatty tissues of the body. But most vitamins are water-soluble vitamins, and they cannot be stored in the body. Rather, this type of vitamins are obtained from food, that are foods that providing these vitamins. For example, thiamine, riboflavin, thyrodoxine, pantothenic acid, niacin, biotin, folic acid, vitamin B12, ascorbic acid, inositol, choline, P-amino benzoic acid, and lipoic acid are water-soluble vitamins. Well, minerals are another biomolecules. Especially these minerals are inorganic compounds which are required small amount for the body. So they are organic molecules as well as they are macronutrients. They are involved in metabolic functions of the body. For instance, iron is needed to make hemoglobin, a red pigment that carries oxygen in the red blood cells. Calcium and phosphorus are very important inorganic molecules because they are components of bone and teeth, and they are involved in muscle and nerve function too. Magnesium is an important inorganic molecule, which is a component of green pigment called chlorophyll. This chlorophyll is important for photosynthesis. Therefore, without magnesium, no photosynthesis. And that means no chlorophyll. Without chlorophyll, no photosynthesis. Without photosynthesis, no food at all. Minerals are cofactors for enzymes because, as already said, enzymes are proteins. All enzymes are proteins, but some enzymes need cofactor for their normal functioning. Cofactor. This cofactor is minerals. Then, when these two molecules join together, the protein part of the enzyme as well as the minerals, where they are joined together, they form a functional enzyme. Previously, we said protein part of enzyme plus coenzymes can give functional enzyme. Here, the coenzymes are vitamins, but here the non-protein part of enzyme plus cofactor, which is mineral, and they give us a functional enzyme. Therefore, vitamins can be used as a coenzymes for the normal functioning of enzymes, as well as minerals can be used as a cofactor for the normal functioning of enzymes. Minerals are obtained from balanced diets that we eat. So a source of minerals are balanced diets of our food. Well, water is the final biomolecule, which is inorganic molecule in nature. It is formed from 2 hydrogen atoms and 1 oxygen atom that are joined together with covalent bonds. It is one of the most plentiful and essential compound in nature. It is found in plentiful and as well as very important for living organisms. Its character, it is tasteless as well as odorless compound. It can be exist as gaseous, liquid as well as solid state. It has ability to dissolve and are used as a medium for transportation of many other substances. Many substances can be dissolved in water as well as they can be transported by carrying through water molecule throughout the bodies of organisms. Since oxygen is higher electronegativity than hydrogen, therefore most of the time electrons are spend their time around oxygen atoms than hydrogen atoms. This means that even if the whole molecule is neutral, but the oxygen side is negatively charged, whereas the hydrogen side is slightly positively charged. The oxygen part is slightly negative charge, whereas the hydrogen side is slightly positive charge. Therefore, water is said to be polar. By the way, this nature, being the oxygen is slightly negative in charge, whereas the hydrogen side is slightly positive charge, will help the water molecule to make hydrogen bond. In the next slide, I will try to show you how a water molecule hydrogen bond is formed in water molecules. Therefore, this is very important characteristics of water for the formation of hydrogen bond in water molecules. To define electronegativity, which is described as ability of atom to attract electron toward itself. When you compare the electronegativity of oxygen and hydrogen, oxygen has higher electronegativity than hydrogen because electrons are most of the time spend their time around oxygen. Here is hydrogen bond, which is formed between two water molecules. The electrostatic attraction between partial positive charge of hydrogen atoms and the partial negative charge of oxygen of water molecules result in the formation of water molecule. To make clear, there are two water molecules here, each of which are made up of one oxygen atom and two hydrogen atoms. The electron, oxygen is partially negative or slightly negative, whereas the hydrogen atoms are slightly positive in charge. Therefore, whenever two water molecules are close to each other, the partial negative charge of oxygen make a hydrogen bond with partial positive charge of hydrogen atom of another water molecule. So in this way, hydrogen bond is formed. Whenever we add another water molecule, immediately the negative charge of oxygen atom of the new water molecule is hydrogen bond with partial positive charge of another water molecule. So in this manner, whenever water molecules are added, there is many hydrogen bonds are formed between them. Many properties of water are directly related with hydrogen bonds. For instance, ice float. We all think that ice is higher denser than liquid water, but it is less denser than liquid water. This is because hydrogen bonds hold water molecules in solid water or in ice far apart to each other than in liquid water. Here, in ice, water molecules are far apart to each other, but in liquid water, they are nearer to each other. This is because of the hydrogen bond. By the way, these properties of water helps organisms in water bodies at least in two ways. First, if I sink into water, physically living organisms which live in water bodies can be damaged, but ice float. Second, ice float because it prevents heat loss from water bodies. Therefore, organisms which live in water bodies stay in constant body temperature. The second property of water is high boiling point. Water has relatively high boiling point. Around 100 degrees centigrade heat is required to change liquid water into vapor. This is because of these hydrogen bonds. In liquid water, to change liquid water into vapor, we need to break all these hydrogen bonds. Therefore, relatively high energy or heat energy is required to break all the hydrogen bonds in liquid water to change the water into water vapor. This is, we say that water has high boiling point. Cohesion is another property of water. It is attraction of water molecules one another. If you think of long trees, water is attracted or absorbed from the soil by the roots of the tree, but photosynthesis occurs in the leaves of the tree. So water is attracted by or absorbed by the root. Then after it passes through xylem tissue and reaches to the leaves for photosynthesis purpose. So one of mechanisms that help water to move up is cohesion. This is a force that helps water to move up from the root to the leaf of a plant. Therefore, water molecules are attracted to each other to move up. So these properties of water, or this property of water, is also related with hydrogen bonds. The other property of water is adhesion, which is the attraction of water molecules with the non-water molecule or with the surface. This is attraction between water molecule and the non-water molecule or with the surface, meaning water moves up from root to the leaves of plant through xylem. Therefore, the xylem wall is attracted with the water molecules. This helps water to move up. For with the cohesion as well as adhesion forces, water can move up in long trees. The other water properties, it is surface tension. This is the water molecules on the surface have the hydrogen bonding only within the liquid, but above water surface there is no water molecules available for hydrogen bonding. This means that molecules at the surface experience innate force that pulling them inward. Therefore, this property helps organism to walk on the water surface. Therefore, this property of water helps organism to walk on the water surface and look for their prey, as well as lighter seeds can move on the water and disperse it elsewhere. So these are different properties of water which are directly related with hydrogen bonding. Thank you all for watching us. Please encourage our effort by like, share, and subscribe our channel. Below are some questions. So if you like, you can do them. Thank you.